Keratin is one of the most important proteins in our body because without it, we wouldn't be able to function properly. Keratin is a fibrous structural protein in our epidermis that provides our hair, skin, and nails with strength and support. In addition to providing structural support, keratin also gives skin its protective properties and serves as a barrier between an organism and its environment to protect the body from pathogens and other toxins that could potentially be very harmful. Keratin also prevents loss of moisture, heat, and other important constituents of the body.
Keratin is essential to our bodies because it determines the strength of our hair, skin, and nails. Excess keratin in the body can cause keratosis pilaris, a skin condition where the skin's hair follicles get clogged. This condition causes entire patches of skin to be covered in small, scaly bumps and rough, dry patches. Most people who have this skin condition says their skin feels comparable to sandpaper. On the other hand, a lack of keratin protein can result in other negative effects. Signs of keratin deficiency include hair loss, premature aging, skin sagging, thinning of hair, and wrinkles. Since these effects tend to be superficial, companies often advertise products that will offer them keratin supplements in order to give them a more youthful appearance. As previously mentioned, keratin is found and produced in the epidermis of the skin, hair, and nails. This protein is produced by a type of cell called keratinocytes, which are found in the deepest basal layer of the stratified epithelium that comprises the epidermis. Keratinocytes actually make up approximately 90% of all cells, making it the most abundant cell type in our bodies. Crazy, right?
In order for our hair, skin, and nails to be strong, keratin first needs to be synthesized. The first step of protein synthesis is initiation. Here, messenger RNA attaches to the small ribosomal subunit. The sequence coding for methionine, AUG, is always the start codon for mRNA. When mRNA reaches the site, the initiation codon binds to the anticodon of tRNA. They are then joined by the large ribosomal subunit. When both of the ribosomal subunits bind together, the A site, P site, and E site are formed. At the A site, the tRNA anticodon binds to its complementary mRNA codon. In elongation, the ribosome moves along the mRNA strand by 3 nucleotides, which results in the transfer of tRNA, where it is released in the A site and then is diffused into the cytoplasm of the cell. As the process of amino acids being added continues, the protein chain gets longer. Termination occurs when the stop codon appears in the active site of the ribosome. Now, our new protein is ready for packaging and export.